Hello, everyone. Thank you for attending today's webinar, How a Sponsored CMC Platform Accelerates mRNA Projects to Clinic, presented by Aldevron. I'm Rebecca Kushmiter. I'll be moderating this webinar. Our speaker today is John Cooper, Senior Director of RNA Sciences and Analytical Development at Aldevron. And joining us later in the Q&A is Bo Chung, Director of Regulatory Affairs and Compliance at Aldevron. All right, let's begin with today's presentation.
Hi, my name is John Cooper, PhD. I'm the Senior Director of RNA Sciences and Analytical Development here at Aldevron. Very excited to be talking to you today about the advances we're making not only in analytics, but also in the processing of mRNA into drug substance and drug product. My career, I have 10 years of public service, first at the VRC as the Analytical Development Lead, building the team, working on protein, DNA, and RNA vaccines. Then also worked at Rare's PBF as the head of development, working on PAV, LAV, and other opportunities for vaccines, and as well as mRNA. And now here at Aldevron, working towards advancing clinic projects faster. So what I'd like to talk to you today about is some of the learnings I've had over my years in the space that I work in, the translational development space, both in analytics and process, and how we can move your project faster with the right information and time. The title today is How Your Sponsored CMC Platform Accelerates mRNA Projects into the Clinic. The most important thing that I find that we need while we're doing development is a starting point. If we don't have an ending point, we don't have a good target, we don't have a good plan, and that means we cannot have a good outcome. The one key aspect of this is having a target product profile from the client that has been approved with the sponsor and their regulatory. The more information on this TPP that is known by us, the developers and the manufacturers, the easier it is for us to hit our targets, not only in assay development, but also our critical quality attributes in process development. The critical target product profile attribute for us is dosing and administration strength. Knowing the amount of material that needs to be into the final drug product is critical, not only for process development and understanding the process through characterization and validation, but also in designing an appropriate analytical panel, as well as acceptance criteria and eventually specifications for release of the drug substance and the drug product. The more information known about the project up front, the easier it's going to be for us to develop and advance it to GMP in the back. In general, mRNA is relatively straightforward and similar, although there are unique challenges based on size, sequence, and type. In general, though, we're looking at a 5' cap, an untranscribed region 5' with a start codon that's encoded for the T7 polymerase that then gives you the coding of the protein, a 3' UTR including the stop codon, and a poly A tail if it's an encoded project. Very straightforward. Not going to dive too much into the technical details of the mRNA itself. We're looking more at process and analytics on here. How we make RNA is similar to how a cell does. It starts with the DNA and does a primary transcript to RNA, including capping at the 5' end. An endonuclease cleaves at the 3' poly A site, followed by poly A polymerase and ATP adding a polyadenylation tail, which is critical. At that point, RNA may be spliced and then transported to the cytoplasm outside to be used for translation to protein. This offers us several opportunities for improvement, not only to the material quality, but also to the process for increasing recovery, expression, and the amount of material that we can then forward process into ultimately drug product. Some of these steps include improving enzymes, modified nucleotides, evaluation of single-use vessels, and continuous flow propagation in the IVT reaction steps. We're also evaluating multiple different types of capping, including co-transcriptional capping, multiplied efficiency, and higher concentration reactions based on our improved enzymes. Tailing, while often preferred to be encoded due to repeatability, is continuing to be enzymatic for gaining longer poly A tails. These are protocols and procedures that we are becoming very good at. Repeatability. Purification of these produced mRNA transcripts includes ion exchange, affinity resins, which help to remove double-stranded RNA, abortive transcripts, and the other reaction components, And with the fact that there's a coated or even an enzymatic tail, enables us to directly target this, analogous to a protein A in an antibody purification. At this point, there's generally a concentration and a buffer exchange into the volume, formulation, and concentration that's needed. And then final fill finish into a container closure for drug substance, or forward processing into lipid nanoparticle drug product with a fill finish. During these, we have several steps of analytics during in-process testing, drug substance release, and drug product release, not to mention incoming materials QC. Each of these assays are following a templated platform approach with iterative improvements as the technology advances and our experience is improved. When a client comes to us with their proposed process, it may or may not be the most efficient one in our minds and in our experience for that product. How we treat this is different based on the risk aversion of the client. Aldevron has been and will continue to improve on our basic platform that generally has a high level of success regardless of the transcript. Client proposals often take aspects of our platform process and have targeted improvements based on their specific transcript. Upfront, we prefer to do a gap analysis on these. We like to do a comparability evaluation of the material that's produced through these two methods so that we can get to the most efficient process, saving not only time, but potentially reducing unit operations, which also reduces risk from additional handling and loss of material through loss of efficiency. To increase the quality of the molecule itself, there are several opportunities for design and optimization of not only the transcript, but also the process, including maximal production, titer, with the optimized CQAs, an increased stability, which would prevent degradation in situ, an increased translation efficiency, and also decreased clearance, which could be synergistic. And obviously a low immunogenicity except towards targeted expression. And now the potential approaches to improve these designs are multifold. We are looking at multiple different enzymes. We are looking at revising and altering the 5' and 3' UTRs and cap structure on the 5' end, including and continuing to modify nucleotides, evaluating alternative vessels and implementing single-use approach at our GMP scale, as well as controlling the length of the poly-A tail through encoding or improved processes during enzymatic tailing reactions. And lastly, by increasing the quality of the transcript through codon or sequence optimization. Essentially, we're looking to balance the intrinsic and extrinsic immunogenic properties of these various molecules. There are several opportunities here through IVT, including optimizing or improving ribonuclease inhibitors, pyrophosphatase, T7 RNA polymerase or alternate polymerase, DNase for template destruction and removal, poly-A, polynuclease in the case of an enzymatic tailing reaction, and also these additional enzymes for capping reactions. Through both targeted mutation and directed evolution, we are looking to improve. We are in the process of improving these enzymes and see continuous improvement occurring over the next several months and continuing throughout time. Once we now have optimized the transcript and we've produced it at a high enough titer to give us the amount of material that's needed for the target product profile, we turn our eyes towards purification strategies. Now, separations are generally the limitation of your efficiency of recovery of your target molecule. And that way, We look to minimize unit operations as well as optimize loading, flow rates, temperatures, and all other critical parameters for any type of purification strategy. We are able to do multiple different types of approaches, both at GMP and in our research area. And we are continuously developing new procedures in our research area to then adopt into GMP. Basically, we see ourselves as having no limitations. It's just work that needs to be done. We have several purification strategies we've been successful with over a large number of projects, including silicon membrane-based purification or bind and elute. We also do flow-through. We have precipitation and centrifugation for tighter assays and for early preparation of research materials. We use various ion exchange chromatography resins. We've leaned into affinity chromatography as well as reverse phase chromatography for reducing dsRNA. And we actively use TFF and filtration as required to buffer exchange, hit the target concentration, and produce material that's forward processable. The second part of having a nice target product profile dives into the analytics and having an analytical target product profile that supports the TPP. Essentially, this allows us to understand the targets or the values that we're looking to aim towards. This is a living document that is expected to change over time as methods and processes improve or are eliminated, or as regulatory expectations change. In this case, there are a large number of attributes that can be considered during this method exchange of the target profile. Essentially, as I mentioned during the target product profile, the concentration of the drug substance and drug product is critical to understand the work range of the methods needed for those release assays. After that, we go through our standard work of identifying and risk assessing various modalities to address the need. We do have a platform set of assays in alignment with the latest guidance, and we look to increase on that knowledge and experience and advance and iterate those methods as we improve our own experience and the capabilities of the technology. Assay development is pretty straightforward. We like to have a very rigid handoff system so that we're not missing critical details, records, or information, so that we do not lose information to a tribal system. We do have a nice system of recording. In this case, when we start with a fresh assay development, we're looking for a literature review, as well as any proof of concept experiments. Literature review is there anything out there? Proof of concept, for example, if it's HPLC, is there a target column, and am I able to see a peak or resolution of peaks of interest? After that, workspace definition and range finding is very critical to understand what the capabilities are of the assay and to start to define what the validation and qualification strategy will be for phase 1 and beyond. At this point, we also include an indication and an identification of the critical reagents, which is part of the control strategy. We want to understand the performance of the reagents over time, as that may have an impact on the data results that is not part of a change in the product. We spend a significant time on optimization, as well as continuously challenging the workspace and the failure at the edges to better define what our range is. We also do evaluation of precision, variance, accuracy, bias, as well as evaluating each method for being stability indicating towards its inclusion on the stability panel as appropriate. At that point, we're looking at robustness, which could be slight differences of the concentration of the reagents, time around the handling, and various parameters that we would discuss during the DOE. Towards the end, we want to have a very clear understanding of what the sample and system suitability criteria will be. Again, critical part of our control strategy. Ideally, including intermediate hold times for the end process as well as the final samples, working hand in hand with the process development group. Towards the end, identification and characterization of those impurities, as well as the main peaks, which I believe we've left out here as the characterization or peak characterization to confirm positively that you're looking and characterizing, quantifying what you believe you are. And then a drafted SOP and a complete development report covering not only the positive results, but also the experiments that were not successful. Internally to Aldevron, we have a lockdown tech transfer process that is overlapping and complementary between multiple different groups that are co-located here. In general, we take products and move them from RNA sciences, which is new RNA materials discovery and development, process design with the client, scale down process development and demonstration if the client's work is needed. Our research grade department produces material that conforms to lab bench or experimental use only material, not for human use. However, the processes and the materials that we use are comparable to those that we use in Tech Ops and GMP. Therefore, we believe the material we're producing in research grade is representative of that produced at Tech Ops and GMP, giving the client an early look into critical quality attributes, again, helping for the control strategy, setting specifications, and understanding the size of batch and other parameters needed to hit the target product profile. Internally, and to me, this is incredibly clear, but I understand this is a quite complicated slide that's actually been cut down quite a bit. Coming to Aldevron, coming to my group for a process, either evaluation, assay evaluation, or to move a product into GMP, we generally start with client-provided project tech transfer documents and information materials. Even better if you have experience in production and analytical data or methods and SOPs. In that case, we would take that into our research grade group, which again is our production research group at a higher scale, and do a demonstration run or familiarization run. Some companies call it a tech transfer run. In this case, the key is to put it at a scale high enough that it will mimic the GMP expectation of process without being too low so that we'd have to use spin filters and non-representative unit operations. We take that material, we would want to evaluate it based on the platform analytical panel. We'll get into what that means in a minute. And it could be either the Aldevron approach or the client approach, right? It's either direct tech transfer from client or it's the platform work from Aldevron. If everything looks great, we've hit our pre-agreed upon targets and all of the assays and unit ops look great compared to the are acceptable compared to the historical experience of the sponsor, of the client, then we would move straight into validation QC and the engineering and GMP runs. If we are not seeing the types of recovery, if we need to further scale up, if we need to add additional unit operations, for example, to remove additional dsRNA or additional endotoxin, then that would be pushed into the R&D or our process development group, which would then develop methods, analytical methods if needed, unit operations as needed, and then further characterize the materials, the methods, and the processes, including generating a development report that describes all of the work that's gone into that. And this is part of a collaborative work with the client in establishing what experiments are required and what data is needed to support not only advancing the manufacturing, but to support the CMC filing for the client. Once we've developed the methods, the process, the unit op, whichever portion or entirety, we then transfer that to the R&D group, which is co-located with the R&D group. And again, at scale, 20 mil scale, we run the developed material and again, run the updated methods And now we have a protocol. You can see again, there's two routes. It either worked glowingly or there's still room for improvement. If there's room for improvement, here we're looking at joint discussion and troubleshooting between the internal groups as well as the client. If it goes forward, we're in business and we're moving towards qualification of the methods, completing the batch records, and moving towards manufacturing. The key here is these are systematic and described handoffs that have timeline restraints as well as templated documents that make the process more efficient and straightforward to fill in, enabling us to provide uniform data to each of our clients in order that they can successfully file their CMC. Our approach is a platform approach. In micro, when I've used platform approach, I equate it to a diving platform. You start there for everything, for the same type of approach. If we have RNA, we would like to put it through these general assays. We do have slightly different assays based on the length and type of RNA, but this is our general panel. I've excluded some of the compendial assays, some of the longer assays, and some of the non-product specific assays that are required. However, looking towards the right here, the key is we've developed this platform so that minimal development is needed, potentially product specific qualification, which again would be minimal, having not done massive development, whether it's a new buffer, whether it's a new concentration. Those are the types of things that we would look at. For potency or cell-based assays, obviously there's going to be some product specific development there, as each product is expected to produce a unique protein. So therefore we would need unique reagents to detect said protein. For the capping and the tailing assays, currently clients are moving into phase 1 with these as characterization assays. But as we improve our resolution, as we improve our characterization and quantitation, I expect these to become pretty standard release assays, and the product specific development here will be expected to drop down even further. For the others, including the impurities, again, minimal development. We're focused mostly on interferences or inhibitions. And then we do the minimal product specific qualification to minimize the timeline, to be the most efficient path, to have these assays available for release in QC. In advance of these assays being available for release in QC, they are available in R&D to be run. Now it's in R&D, so it's not under the quality system at that case. It's done with high quality, but does not fit into GMP testing. It may be used for characterization or at least for helping to set specifications, but not for, I would not suggest it for comparability or for release. We have the ability also to test these methods in our validation department, which in that case, the validation department does have qualified instruments. Those assays are qualified, obviously, by the validation department. Those would be GMP usable, and that is the path forward for additional data that is not part of the standard platform of assays. One other thing to look here is we have multiple assays listed, or multiple types of technologies listed for each assay is what I should say. And I've mentioned getting to phase 1 to line up with the USP guidance. We're doing capping efficiency. I'll show you quick data on that one, as well as poly A tail. We have also implemented mass spectrometry for characterization of peaks, not for release at this point. And my personal opinion is the further we can keep mass spec away from release, the easier it will be in QC. However, that is not a hard and fast statement. As we move to impurities, we're looking at product and process related impurities. Typically right now, we're very interested in dsRNA and the reduction of that. We are working on multiple different avenues to increase our sensitivity for that assay as our processes improve. Further, we're increasing our specificity on the process related impurities. Nano orange captures all protein. However, enzymatic specific ELISAs, whether used for release or process development, will help us understand better which components are being selectively retained in the product. Mass spec is a strong tool and gives us generally an unambiguous answer. However, it is not practical to deploy on a regular basis in QC. Therefore, we rely heavily on it in the development space as a characterization support for the peaks in the assay, for the generation of C of A for the quality of a reference material or interim reference standard, and for any troubleshooting and confirmation that may be needed. Briefly, the idea being this is an RNA size and purity assay, which is all based on electrophoresis. We see 3 different types of core instrumentation based on this technology. Tape station by Agilent. We have the fragment analyzer by Agilent, and then the general, most well-known, the denaturing agarose gel electrophoresis. Now, unfortunately, these are not all the same project, so it's not an apples to apples comparison. But what I'd like to show here is not only do you get different types of data from different assays, but in all of these views, the resolution is limited based on the resolution gained in the gel. The tape station itself is a very small, very short flow path. Fragment analyzer has a little bit better, but maybe not quite as good sensitivity. And gel electrophoresis has a short path length and a very narrow range of detection. Along with the streaking, it's very difficult to assign specific peaks as you would with a chromatogram. The advantage of the tape station and the fragment analyzer is we can look at chromatogram or electropherogram based views and potentially pull more information from that. However, because of the lack of resolution, it's not an advantage. Moving towards SciEx capillary electrophoresis instruments and the lengths of capillaries that are allowed in those systems, that are enabled in those systems, the quality of the coating on those capillaries, and the repeatability and range by which those kits and instruments work provide me with high expectations for good performance in the future on those assays, especially having seen preliminary data that I hope to share at some point here in the future. Our current capping assay is based on UPLC and is supported, as I mentioned, by mass spec peak characterization. In this case, we have one mRNA project that has been capped and gone through full processing. We have a second sample that we have excluded the capping step, meaning that it should be 0% capped. I can't go into too much detail about the amount of work that went behind this, but to have this type of baseline resolution, which clearly shows capped and uncapped in the same chromatogram, baseline resolved, or at least nearly baseline resolved, to give us a very good understanding of percentage. And as you'll notice on both the left and the right, these are the calibration curves essentially that arise from adding more and more and more of each of those. So a proportional increase in the amount of material of capped results in a proportional amount of signal. Same with the capped. In this case, incredibly encouraging. Anything above 0.99 to me is an incredibly strong assay, and we look forward to establishing this on multiple different programs. Size and purity is generally important. Most poly-A tails now are being encoded, and we try to encourage encoding because the length are more efficient and uniform. However, they may not be as long as an encoded tail or as an enzymatic tail. So there may be an opportunity to combine those two. Aldevron is a method that focuses primarily on the relative length of the poly-A by comparison to poly-A fragments, calibrators of the known length. A very straightforward assay in that case. Moving towards CE, we're looking towards not even needing the ability potentially to do a digestion in advance of resolution of the poly A tail, as well as other approaches. In our current experience, clients are more interested in reporting relative size of the tail. However, our assay, as all of our assays, are amendable to be used for additional attributes on top of those called out. As Aldevron and other opcos under the Danaher umbrella work towards establishment of a beginning to end process for producing LNP encapsulated drug product RNA, we're aligning our drug product panel with ASTM guidance. These assays, again, will meet the current platform expectations of the FDA, and we are currently iterating the technology and improving on our ability to analyze these materials, and better yet, to understand the meaning of the data to help assign which is a CQA and which is not. In this case, looking at lipid nanoparticles requires you to evaluate both the intact particle as well as a disrupted particle. So there is a slight addition to the amount of product-specific development that's needed here. However, as we come to a platform or a library of LNPs and our experience in analysis, those SOPs will be in place, and the product-specific development will be limited to conformational experiments in most cases. We like to talk about qualification and validation in this space, and I'm not going to spend too much time here because it really depends on which phase you're looking at. A majority of the folks that are coming through our process, working first with RG material, research grade material, and then looking to develop a process and move into GMP for phase one, are really only needing qualification. Validation hits when you're moving into phase three, late phase three, and commercial, and the statistics require a much higher N to be accepted. These words are often used interchangeably. I like to call what we do qualification or small v validation. However, there are several parameters that are needed to know regardless of how we call it. And for us, the validation acceptance criteria and the fact the method is suitable for testing and release of the GMP material, again, based on the TPP, are the critical things to focus. Without spending too much time on the goals of the validation and qualification, the key here is that we have a flexible approach based on each individual client's projects, both on the phase that they're in and status of the process and product, as well as the type of product and composition of the LNP and formulation. Regardless of construct, regardless of modality, really, these are the critical attributes that are evaluated in a qualification study. To summarize our experience with process and assay development, I'd like to make several statements that I believe to be incredibly true, as well as building in confidence and experience as days go on. We at Aldevron are committed to safety and appropriately advancing sponsored programs efficiently into the clinic. We want to move fast, but without haste. We want to do it correctly and completely and speedily. The continued expansion of our project management features enables these efficient timelines because they help us stay on track. Scientists are scientists, but without having a project manager helping us with timelines, we can tend to fall off into a rabbit hole of something of interest rather than something of critical need. Having project management, having our teams co-located, having our governance listed clearly, and our goals and targets aligned upon enables us to do this in a much more efficient manner. And having project management coordinate on top of that, allows the scientists to do the science and engage with the clients on the appropriate timelines. A major concept I'd like to walk away from here with is that the client-sponsor definition of the target product profile and their contribution towards defining the analytical target product profile are absolutely critical to the success, efficiency, and timeline of process and analytical development. The better the target we have to shoot at, the more likely we are to hit it. Aim small, miss small. Development of our assays and our manufacturing unit ops is completed as phase appropriate and fit for purpose after completion of the gap analysis and in conjunction with the client. We are the providers of the information. We want you to be successful. Your success is our success. Standard platform assays have been instituted as a panel. I call these platforms, again, these are not the assay. These are the fairly well developed, might actually work as it is. At the very worst, we should need a week, 2 weeks potentially of development to lock it down to a place where we feel comfortable collecting data to establish acceptance criteria and support specifications for your projects. And a core concept from Danaher, and one that I know that myself and the team we put together here believes, is continuous improvement is critical. Selfishly, it's critical to the development of our staff because we're interested in learning more, doing better, and improving. And it's critical to our clients because as we advance technologies and improve the assay panel, we are going to be more efficient, we're going to have better information, and therefore a higher level of success. With that, I'd like to thank you for your time today, and we really look forward to any of your questions. If you are interested in speaking with us about product development or product manufacturing, we look forward to hearing from you. If there is analytics involved, analytical transfer, process development, or even CMC, I look forward to meeting you and having those discussions. I think you're going to like our team, and I know that our team will do good work. Thank you.
All right, let's move on to the Q&A. What are some of the most important considerations of an analytical testing panel when developing an mRNA drug project?
That's great. And I can take that one because it is a little bit variable based on the exact type of project and the size of the trials that you're looking at. But the key quality attributes are what we typically go after on top of the general panel. And product experience by the developer really helps to drive and define that. Now, if we're doing product development, we're the ones that are going to pop out those CQAs and understand what those critical holdups would be. But the more that the client and sponsor know coming in, the easier it is to get to those answers.
Great. Our next question here. What support is available to help developers through the product design and development process?
Oh, that one's also for me. I've prided myself in my career of being an educator of not only my staffs that I've built, but also of the clients that come work with us. Often we're working with smaller clients that have not gone through this process before, or it could be their first time even at a larger company. And since we have 20 years of experience of going through that, we can provide our opinions and our histories of learning and provide options that we believe are going to go forward. And with our alignment with our regulatory and our communicated platforms and continued success in the filings with the FDA for various clients, we feel confident that that approach is correct and that it'll be appropriate for anyone that comes to us. Flexible, agile, and we're going to try to hit it quick.
Great. I think the next question, how do you ensure efficient tech transfer for an RNA program? What should be prepared for a meeting with a sponsor?
Yeah, I can take that one. And John actually hit on this. in the presentation. Really, the key is to understand your TPP and align with the regulatory agencies. Communicate with the agencies early and often to align on your development. Coming in with that understanding allows our team here and the development of the methods to go much more smoothly because, like John had shared again earlier, understanding your end goal in mind early on lets you get that that work in quickly and early.
And then any gaps would immediately, I would be engaged in that program, looking at what the information is that I don't have to move forward. Again, my group and my teams are all interested in being successful for themselves and for the company and for your company. So the easier it is for us to start, and that means if we have to dig in and start asking questions, then we'll have more meetings with you. But at the end of the game, we want to ask for permission before we proceed rather than coming back later and saying, whoops, this is what we did. Please forgive us.
All right. Next question. To carefully develop and manage plans for a clinical program, how can developers effectively balance pressures of time and budget to hit clinical milestones?
Oh man, that's the golden answer, right? So really, what we offer is, and again, I've been in the field long enough to know that there is no perfect. There's only progress. So the increase in communication up front, transparency in communication, sharing information, and like I said before, I'm someone that leads my teams by and ask for permission rather than ask for forgiveness. So the earlier we discuss and align up front on what we need to do, and then bring in regulatory and the sponsor to make sure that everyone's on the same page, we go through it once and we do it correctly and we move on.
Great. Another question here. With long lead times in manufacturing, what recommendations are available to help shorten delivery of products?
So again, the easier projects to advance are the ones that have more information up front. It doesn't necessarily mean that you've made 10 batches of it and have 400 different preclinical trials. It means that you know what your target product profile is, including concentration, critical quality attributes. So the better that that program is defined, even before it gets to us, the quicker it's going to move through, because now it becomes standard work rather than baseline, starting from the baseline and working your way up. So we want to leverage the existing knowledge. We don't like to start from scratch. We like to do confirmatory, and we like to advance projects at an appropriate speed.
Wonderful. Our next question. Can you elaborate on the support that Aldevron provides for developers in CMC filing?
Yes, we have supported many of our clients really at all stages of development, from early pre-IND meetings through their IND. We've supported our clients with filing the CMC portion in their CTAs for ex-US jurisdictions, all the way through late stage BLAs, marketing authorization, and have supported some clients now with supplemental BLAs. Um, with their indication, and especially with infectious disease or the seasonal flu and so forth. We support our clients. The majority of our support is with respect to the drug substance portion and the, the the regulatory strategy on how to file for your critical starting materials. There's a couple ways we've seen different sponsors approach that. Um, Additionally, we have supported our clients with the regulatory filings for even their drug product. We may not, even manufacture that for their program specifically, but given the source data, we've successfully filed a number of those projects with their clients. So really, in short, it's what our clients need. It's what you need. We're happy to provide that support.
And even further, as far as the analytical panel goes and the process development work goes, again, I've always leaned into including CMC type write-ups directly into the SOPs, that the SOP is now directly aligned with what we're communicating to the FDA. So that information is essentially that well, really it'd be PIND information is available as soon as you engage us.
All right. Our next question here. What have we learned about the manufacturing, analytics, and regulatory due to the pandemic?
Yeah, I can maybe begin with this question here. John has shared with the team here the analytics aspect of it and leading into how that is complementary and supportive of your manufacturing process development as well. From the regulatory perspective, in addition to what John has already mentioned, we see very big emphasis on the starting material with the linear template, having that be a high quality input in addition to the nucleotides, the enzymes that's all that's used in the mRNA manufacturing process. So a lot of regulatory scrutiny coming that. So in short, ensuring that the input is high quality, the process is well controlled, and that we have the analytics to understand the release, the output of what we manufacture.
Okay. The next question, are you able to discuss target project parameters that include intermediate and API storage, formulation approach, endotoxin removal, or lack of production?
Yes, but not in this setting. So we have a significant amount of work that is still considered trade secret that would be employed on the projects. We're in the process of protecting it, but we are employing it at the same time. So the answer is we have a very, what we believe to be a defensible position that we're moving into. And then as we iterate, it will become more and more differentiating. We feel that we're moving towards best in class in this field, and it's full steam ahead.
All right. Our next question, are there any innovations that can be suggested to help shorten lead times or overcome current challenges?
Absolutely. We are an innovation-based lab, our development facility here. And so we're always looking for not only processing projects through that are relatively straightforward, but we're looking and really I'd argue my team would rather have the more challenging projects that we're able to learn and expand our knowledge and expand the availability of the field. So the innovations, if you have ideas, thoughts, especially if there's data behind it, excellent to be shared. And then as far as shortening lead times and overcoming the current challenges, I can only reiterate the better the target product profile and the analytical target profile are before we engage, the faster we're going to get the right answers.
All right. And I just remind you that you can refresh your screen and line up your audio a little better. I am hearing a few echoes as we go through these questions. Give you a moment for that. All right. Our next question. Are you aware that FDA favors particular plasmid for in vitro transcription on mRNA products?
Yeah, so if I can seek more clarification from the question there, so we have typically what we see are the linearized DNA templates, the constructs, and the stability of that construct is designed by our sponsors and early on in R&D. John, maybe is this something you can address or elaborate on?
I'm sorry, at that point I was reading something else.
Oh.
Let me see if I can go back to the question here.
Yep.
FDA favors plasmid for in vitro transcription. Oh, of course, they're going to favor. Absolutely. I'm not sure there's a specific identity, but there's definitely the work and the documentation and the non-animal origin behind it. So I don't know if that's a quality level question, but certainly everybody wants to do everything the absolute correct way because we are talking about patients in these cases. So we are aligned with the FDA and they're improving or iterating guidance, and we work to follow those. We have teams within Aldevron that are dedicated to reviewing and understanding and communicating and disseminating that guidance to us so that we are aligned before we even engage the client. So yes.
Okay. The next question. When we set up the minimal residual dsRNA as priority of process development, what is the recommended downstream process?
So that was actually the one I was reading. And in our case, we do have a residual template removal step, and that's really about it. And so that one, not that I'm saying that that's infallible, but that's where we would start. And the downstream from that does follow on. Our clearance is several logs above what's needed, and we could certainly characterize that for each individual project.
Okay, I think this will be our final question. We have had a lot of great questions today. We were not able to get to them all, but we'll try to get back to everyone who submitted personally after the webinar. And to wrap it up, the question is, how do you control dsRNA for your processes, and what levels are typically expected in your process?
So there's a few different things. It's hard to talk about every single project because of the wide range of sizes and attributes that they have, and the different variances in how those happen. We control for dsRNA through a few different ways, not only in updating our enzymatics, but also in improving our processes, not only in the IVT reactions, but also in the clearance unit operations. We do have two different unit operations we're putting in place to directly address this. And then again, as I mentioned earlier, we are working on multiple different protein enzymes to directly eliminate this during the IVT step. So seemingly everything we're targeting here, we have multiple parallel paths, and that's by definition. Get down those paths as quickly as we can, pick one winner, and then really hit that one hard so we can start implementation quickly.
All right, well, thank you so much. Thank you all for attending this Fierce Pharma webinar and submitting so many great questions. I'd like to thank our speakers for participating and Aldevron for presenting today's webinar. This webinar has been recorded. You'll be able to access the recording within 24 hours using the same audience link that was provided to you earlier. Thank you again for joining, and we look forward to seeing you at future events.